Hello, Dan Guerra here from VerEvMed Studios in the magnificent and beautiful inland Northwest. Sorry, I just got in from outside, so that's why I had my prescription sunglasses on. Today, we're going to talk about a subject which I've been meaning to get back to for some time. It involves, of course, the biology and the biochemistry of lipids, particularly lipids that are involved in um, aspects of metabolism and control over that metabolism and ultimately signal transduction cascades. So today we're going to talk about a particular eicosanoid from the lipoxygenase family known as LTB4 or leukotriene B4. This is a VerEvMed lecture. Today is no longer the 23rd of August. It is now, that was when that picture was taken actually, it is now the 6th of September, 2018. It's actually the day my father would have had his 94th birthday. So I'm thinking about him. I'm going to talk to you today about proximal and distal phosphorylation of a lipid receptor which mediates an innate immune inflammatory response. And that indeed will be the LTB axis with its receptor, the BLT. So that's what we're going to get involved in. Again, this is VerEvMed. I'm the chief scientific officer, co-owner of the company. If you have not learned already what we do, we consult with professionals and with lay people on various topics in the biomedical, biochemical, biophysical literature. What we do is we take in clients who ask specific questions related to, say, a given drug interaction or a given physiological response or nutritional component or perhaps even a pharmaceutical interaction. And what we try to do is go to the scientific literature, find the most pertinent papers and the most recent ones, discuss them with our clients at the level that they feel comfortable with, and then come to some kind of conclusions related to whether or not the evidence that is found in the current publications give on a given topic can be verified by looking at the whole corpus of literature going back many years sometimes. And the more elaborate we carry out that sort of in-depth deductive reasoning aspect towards scientific literature, the more detailed we can then provide for our client a complete understanding of the topic that they initially had some interest in. So that's what we do. We're an online company and we're at verevmed.com or www.verevmed.com is shown there. That's my Facebook page. Facebook page is going to have all of these lectures, which are now in YouTube video format, as well as a lot of other information regarding VerEvMed and also some philosophy, because I also write a fair amount of philosophical narratives, mostly Western philosophy. And you'll see that sometimes come up into the topics that I talk about. Like, for example, most recent discussions were on the diaventhology of ankylosing spondylitis, which had to do with logical progression of understanding of how that disease functions at the immune level. All right, so that's me. I'm Dr. Daniel Guerra, and that is my particular and specific email address. You can contact me there. I will get back to you usually within 24 hours. Or you can go directly to the website where the company is and contact us through there. There's a form. All right, that's our logo. I think that's all I really need to say about this page. Let's get going. Get rid of that. All right. Let's talk a little bit about what LTB4 is and what it isn't. The LTB4 stands for leukotriene B4. It's the best characterized leukotriene. We're directly responsible for the presentation of acute inflammation. So it's a local or autocoid hormone, which is intimately linked to the inflammatory response. It activates and enhances the longevity of circulating leukocytes, both lymphocytes and things like neutrophils and macrophages, as well as dendritic cells. Stimulates leukocyte adhesion to vascular endothelium by upregulating integrin expression. It's also present at high levels in synovial fluid and in the blood of people suffering from rheumatoid arthritis, which is, of course, an autoimmune disease. LTB4 antagonism inhibits inflammatory arthritis, although in a recent phase 2 clinical trial, an LTB4 receptor antagonist did not remit the disease. So there's still a lot of medical research to be done with LTB. It has a lot to do really with the kinds of receptors it binds to, whether or not you're going to have any kind of dynamic penetrance of drugs directed towards that axis and them having a positive result. So it's a powerful stimulator of interleukin-6 production by monocytes. That's pro-inflammation. At nanomolar levels, LTB4 induces a 5-fold increase in interleukin-6 transcription and a 3-fold extension of the messenger RNA stability. And it's dependent on a tyrosine kinase activation intracellularly, and that activity is dependent upon the transcription factor NF-kappa B and of the NF-IL6 family. LTB can also activate cytokine-producing T cells. So you've got T cells as well as you've got innate cells, you see. And that's via the LTB4-mediated interleukin-1, another pro-inflammatory cytokine production. And that's after interaction with macrophages and dendritic cells and their secretion of interferon gamma. TNF-alpha, of course, mediates the dendritic cell and neutrophils, and that then engages macrophage cytotoxicity. LTB4 markedly enhances cytotoxicity mediated by dendritic cells and by neutrophils and by monocytes. Indeed, the presence of LTB4 enhances tumor necrosis factor alpha secretion, and the lipoxygenase inhibitors diminish that production. LTB4 is synthesized, if we get into the biochemistry of it here in a moment, but just to give you an initial blush of it, via the phospholipase A2 activity in the membrane, and then the lipoxygenase 5. So there are 3 different lipoxygenases, 5, 12, and 15. It's the LOX5 that is responsible for the LTB4 production. All right, so let's take a look at tumor necrosis factor alpha here. It's, of course, one of the principal mediators of acute inflammation, so it's no surprise it's going to be linked to LTB4. Major source of it is, of course, macrophage. LPS is a potent stimulator, and that's, of course, through a toll-like receptor, something we talked about many times before in these broadcasts. The major effect is the activation of the pro-inflammatory NF-kappa B, which is a transcription factor. Major actions, it increases the endothelial expression of selectins and integrins, which are involved in cell adhesion. It increases chemokine production in endothelial cells and macrophages, so it's involved in the sway and movement and transport of cells towards a site of inflammation, chemotaxis. It increases cyclooxygenase, which is another type of oxygenation of 20-carbon fatty acids. And that oxygenation pathway, or so-called icosanoid pathway, we'll see in a moment, basically involves the incorporation of molecular oxygen to preformed polyunsaturated fatty acids. Most common of which is arachidonic acid, which is an omega-6. It's 20 carbons long with 4 double bonds. Therefore, it's involved in the production of prostaglandins, and when the lipoxygenase functioning occurs, which is a different enzyme and a different enzyme mechanism, but still the incorporation of molecular oxygen into those fatty acids, you generate what are called leukotrienes, and they have various names. And they're also chemotactic, of course. And the one we're going to talk about today in particular is. So you get this whole processing working here. Let's put on not the sunglasses, but these fancy glasses here. All right. Tumor necrosis factor binds to its receptor. It signals through various proteins that are linked to it. This whole assemblage of proteins, IKK beta, many of them dimerize. You have dimers of all of these cofactor adjacent proteins, ones that then adapt to this entire matrix of this receptor. Ultimately, this leads to the phosphorylation of I-kappa B and NF-kappa B, and you get NF-kappa B activation, and then from that transcriptome, you get inflammatory genes being transcribed and then ultimately translated. So there's a phosphorylation cascade involved in this pathway. And you can see how these adapter moieties are necessary for the entire functioning of it. Okay, and you can see that there's a lot of other things that's going on when you induce the tumor necrosis factor pathway, including the prevention of apoptosis and actually an increase in the JAK-STAT pathway and the JNK pathway, and that could actually lead to then proliferation of lymphocytes and leukocytes. Okay, so that's what that structure says. TNF alpha, which is linked to our LKB4, the story for today, induces prostacyclin expression. That is a cyclooxygenase product. And that happens in the endothelium, and that increases local blood flow. Okay, it acts as a vasodilator, prostacyclins do. It increases IL-1 production in macrophages. That's pro-inflammation, of course. It induces apoptosis in some cells and not in others, you see. And that's through the P55 receptor where it activates the caspase, and it's shown up here, actually. Okay. It's a composite drawing showing you all the possible fates that could happen once TNF binds and how the leukotrienes are going to be involved in this. We'll see in a moment. It induces apoptosis in some cells, as I said, at high concentrations in blood. It can induce septic shock, organ failure, thrombosis, and it actually can kill. Now, here we're taking a look at how bradykinin will bind. We'll talk about what bradykinin does in a moment. Binds to its receptor, which then activates a G protein. That activation of G protein then turns on phospholipase A2. Phospholipase A2 then hydrolyzes phospholipids of the glycerol phosphate backbone. A2 because it's removing the fatty acid at the 2 position of the glycerol backbone. That generates usually, because the fatty acid that is found in the 2 position, which is normally residing there, is arachidonate. Once you produce arachidonic acid, you also make a lysophospholipid, which can go on further to signal. We're following now just the arachidonic acid cascade, and we're following the 5-LOX pathway. The FLAP is actually the 5-lipoxygenase activator protein, so it's necessary for this enzyme to function. And indeed, it's showing you here initial stimulation in the plasma membrane, but the actual 5-lipoxygenase, the FLAP protein, and the entire further activity of the LOX enzymes is believed to happen actually in the nuclear envelope and the ER, but mostly the nuclear envelope. So it's a very exotic synthesis of these autocoid icosanoids, particularly of the leukotriene family, because the LOX are found in the nuclear envelope membrane. All right, anyway, so we're looking at 5-hydroxy, hydroperoxy, icosahexatetraenoic acid, to a 5-hydroxy icosahexatetraenoic acid. Then you generate this epoxide, which has a conjugated triene system. So, of course, to be confusing, there are actually 4 double bonds in the molecule, but you have a conjugated triene system. That's why they're called leukotrienes, because they're found in leukocytes, circulating leukocytes, you see. Anyways, you quickly hydrolyze that epoxide and you make LTB4. And we'll show you some more of that activity in a moment. But also, LTA4 can spontaneously combine with, if you have an LTC4 synthase enzyme, you need an enzyme for this, the tripeptide glutathione. So now you're making peptidoleukotrienes, and that's this pathway. First LTC4, then you remove glutamate, you make LTD4, then you remove glycine, and you get LTE4. And the LTE4, of course, is just going to be the cysteadyl leukotriene, right? Because you remember that the glutathione has glutamate, glycine, and cysteine. So there's a self SH group in this particular peptidoleukotriene. All of these are highly potent, and whether or not you go through the LTC4 synthase pathway or the hydrolyase pathway really depends on the cell type. So lots of cells associated with the lung go through this pathway, and in fact, these compounds are very important in asthma and in the terrible vasoconstriction that can occur with problems of lung diseases, whereas the LTB synthesis pathway is primarily in leukocytes and lymphocytes. Okay, so that's the differentiation, is whether or not it has the hydrolase enzyme expressed or the LTC synthase expressed. That's what really decides the fate of the first epoxide residue coming from the LOX pathway. Again, flap is necessary for this whole enzyme mechanism work. And actually, it's interesting. It works through a iron 3, iron 2 redox system, and it also requires ICOSA hydroperoxide fatty acids to activate that moiety. It actually stabilizes the iron 3 level of oxidation so that it can carry out this oxygenation of this fatty acid. So it's a really interesting enzyme mechanism, which sometime maybe we'll talk about. All right. So now notice here we showed you that bradykinin is binding to this receptor working through this G protein. Why is that important? Well, several episodes ago on VerEvMed, I talked about the bradykinin pain transduction pathway. Remember pain transduction? Of course, yeah, lipoxygenase and cyclooxygenase products are going to be involved in the signaling of pain, right? Because some of these compounds are going to be very potent vasoconstricting, and they're going to signal pain at the local level, at the place of lesion, right? That's what this is about. So pain transduction is a process of converting painful stimuli to neuronal action potentials at the sensory level. Excuse me. You have nociceptors, which are free nerve endings, which are receptors which then transduce pain, right? Or transduce noxious stimuli into action potentials, which we receive as pain. There are chemical mediators which alter the membrane potential of the pain receptor. Those include plain old potassium and protons, lactate, histamine, serotonin, the bradykinins, we just saw that transduction pathway last slide, and the prostaglandins, okay, and also, in fact, the leukotrienes. Non-steroidal anti-inflammatory drugs actually prevent prostaglandin production by inhibiting the action of COX, or cyclooxygenase, and therefore you don't produce prostaglandins. The prostaglandins are another way of getting the pain transduction. Prostaglandins are another way of making highly potent pro-inflammatory cytokines. So we're not going to talk about the prostaglandin synthase pathway. We're talking about the lipoxygenase pathway. I'm just showing you that they are co-occurring depending on cell type, and they can switch back and forth. Just to finish off this pain thing so that you get an idea that pain is also associated with leukotriene biosynthesis. When peripheral tissue is damaged, primary afferent neurons are sensitized and or they're activated by things like heat, by mechanical injury, and or by chemical stimuli, which we just talked about. Prostanoids can do it. That is, prostanoids produced in other cells, protons, bradykinin, cytokines, of course. And that all leads to an opening of a cation channel in the neuronal membrane. For example, the TRPV1 is a major cation channel in the neuronal membrane. That will produce an inward current of sodium and calcium ions into the peripheral nociceptor terminal. If that depolarizing current is sufficient to activate voltage-gated sodium channels, such as the NaV 1.8, they will open, further depolarizing that membrane, that now activated membrane via this action potential. That initiates the action potential and then is conducted along a sensory axon to the dorsal horn of the spinal cord. This is pain transmission after the transduction. Central terminals of the nociceptors contain excitatory transmitters such as glutamate and substance P for pain. These activate postsynaptic NMDA, neurokinin, and other receptors. Repeated nociceptor stimulation can synthesize both peripheral and central neurons, and the activity-dependent plasticity wind-up aspect of transmission. Later, sensitization can be sustained by changes in the expression of genes, so the entire transcriptional activity. And those genes have neuropeptides, neurotransmitters, more ion channel receptors, signaling molecules in the nociceptors. Spinal neurons are activated, and our glial cells in the central nervous system. In addition, physical rearrangement of neuronal circuits by apoptosis, nerve growth, and sprouting of dendritic spines occurs in the peripheral and in the CNS. Both induction and maintenance of central sensitization are considered critically dependent on the peripheral drive by nociceptors, indicating there are therapeutic interventions which may work at this particular axis, something I talked about before. So what are the origins of the signal transduction pathway? You have a ligand-bound G protein-coupled receptor. And that's a protein conformational shift that will occur once a ligand binds. That stimulates the dissociation of that GPCR, which is a heterotrimer. the now distinct G alpha subunit and the G beta gamma subunits, which stick together, individually and specifically interact with a unique ensemble of effector proteins and their intracellular secondary messengers. So this is a signal transduction pathway that we're going to be following for LTB4. Okay. Remember, this can be initiated by things like TNF alpha, bradykinin, or just the movement of ions across that membrane. Okay. Eventually, the resultant cellular response to any future stimuli becomes repealed, because you have to, as soon as you initiate a phase like this, where you're going to get a tremendous amount of transduction that's going to ultimately lead to inflammation, you have to have a built-in system. It has to be within the molecularity of the system at the get-go to start putting the brakes on. If you don't do that, you have rampant pro-inflammation, just going to kill all the cells, damage tissue, and can lead to morbidity and ultimately even mortality, particularly if it's in a solid organ or if it's in something like the central nervous system, right? Yes. Anyways, to repeal that activation, it's a process we call repealing it or attenuating. We call that desensitization. And that's going to be kind of important for the paradigmatic system we're looking at today with LTB4. You'll see what I mean. Besides this desensitization, further elaborating phosphoryl patterning, that means adding phosphate to proteins like receptors proteins, and they're via unique protein kinases, which we'll talk a little bit about, further expand the functional repertoire of the GPCRs. Okay, so when you ornament with phosphate and you do it in a temporal and hierarchical manner, temporal meaning in a time course manner, and hierarchical meaning depending on which particular amino acid residues, in this case we're going to talk about serine threonine, they have hydroxyl groups, recall, and they could be easily phosphorylated and the phosphate can be removed. There's going to be a hierarchy of specific phosphorylation sites on this LTB4 receptor, which is going to be working through the G protein coupled receptor system. And that hierarchy of phosphorylation patterning is going to allow for chemotaxis and degranulation of particular leukocytes at the site of inflammation, as we're going to see. That's what the whole paper is going to be about. So ultimately, the disambiguation of these hierarchical kinase cascades, remember those are enzymes that add phosphate, will require very precise multimodal experimental design, which they hope to be able to do. Now, back to LTB4. It is an oxygenated arachidonic acid autocoid, so it's a compound that can be used in the cell it's made in. It can also be paracoid or paracrine activity. It generates within the plasma membrane, from which it can signal externally by binding to cellular receptors found on other cells like circulating leukocytes. Once bound to this receptor, LTB4 becomes a potent chemoattractant for neutrophils and lymphocytes like T cells. Neutrophil swarming, it's called, and it is innate immune surveillance response. Okay, and it is concentration dependent according to LTB4. So the LTB4 sets up a chemical gradient, okay, within the plasma as it's being secreted and building up in concentration, along with associated cells like the neutrophils. They're going to generate a swarming activity of more neutrophils, right? So it's a chemotactic response, remember. So that biochemical gradient decreases in potency according to diffusion coefficients, as you might guess, diffusion of the LTB. And also the miscibility of this oxygenated fatty acid. And that's, of course, all dictated by the hydrophobicity and hydrophilicity of that LTB4 moiety. You also get potentiating binding to proteins, such as that flat protein we talked about. In fact, that's what that flat protein does. Present LTA, for example, to the lipoxygenase. And potentiating binding to proteins, or, of course, degradation, further degradation via enzymatic hydrolysis usually, removing some of those double bonds and opening up that structure a little bit so that it can be further eliminated. Okay, but that does not follow a measured and predictable inverse square law. It's not simply you're not simply dividing by in the denominator, that is looking at the square of the distance from the source of the production of leukotriene B4. It's more complicated than that. It's complicated because of all these different intermediates and cofactors, including the receptor itself. Remember that LTB4 is a lipid mediator. It's a lipid. Okay, it's an oxygenated fatty acid. Chemistry dictates to some degree its availability. Okay, where you're going to find arachidonic acid, which is a precursor, or in fact, the phospholipid is the parent compound precursor. You have to have transport that's all superimposed upon that chemical gradient. That's why it's not a simple inverse square law. People who study physics and think about things like how potency is transduced just according to distance and time, they're working in the playpen. In biochemistry, it's much more complicated than that. So eicosanoids are a special class of lipid mediators. They're highly modified oxygenated chemistries. LTB4 is a leukotriene found in leukocytes, has that conjugated trienoic system. It also has two free hydroxyl groups, making it somewhat more soluble in aqueous than if it did not have a conjugated triene and dihydroxy chemistry. So it's a grand total actually of 4 levels of unsaturated because unsaturation. So there are actually 4 double bonds. It's just you have the conjugation of the 3 double bonds. That's why they're called leukotrienes. All right. Now, let's take a little bit of a look at what goes on when you have double bonds. First of all, you get electrophilic addition. So the pi bond you're looking at here, above and below where the double bond is, okay? There's the sigma bond, which is held very tightly between these 2 carbon atoms. The pi bond is localized above and below that carbon-carbon sigma bond. So the pi electrons are relatively far away from the nuclei, are therefore loose, much more loosely bound, okay? So an electrophile will attract those electrons and can pull them away to form a new bond, okay? This leaves 1 carbon with only 3 bonds and therefore a positive, I love the way that's written there, charge. We call that a carbocation. Okay, so that's right there. So you've erupted now a carbocation because you have an empty p orbital because of the fact that you've made this reaction with this electrophile, see? Tying up those electrons, making this only have 3 bonds rather than 4. So the double bond then acts as a nucleophile, which means it attracts the electrophile. Okay. Most cases, the cation produced will react with another nucleophile. See here? Another nucleophile to produce the final overall electrophilic addition reaction, which is shown here. So you're getting rid of that double bond, but you have to first carry out this electrophilic addition, right? Then you're going to get this nucleophile, again, attacking the electrophile to fully remove that double bond. And now you've got substitution of these 2 moieties here, whatever they happen to be. So you have electrophilic addition. That's probably the most common reaction of all alkenes. And these fatty acids we're talking about are alkenes. Alkene just means it has a double bond. Carbon-carbon double bond, sorry. So here is the parent compound. You notice it's a 20 carbon. That's a fully saturated fatty acid. We call that prostanoic acid, first isolated in the prostate. What happens again? The signal transduction, which involves that pain we talked about, but also this chemotaxis degranulation inflammatory response. First of all, you start with a membrane-associated phospholipid. It can be phosphatidylcholine, phosphatidylinositol, even phosphatidylethylamine. Phospholipid is acted upon by phospholipase A2, PLA2, which can be inhibited by anti-inflammatory steroids like NSAIDs, okay? And by anti-inflammatory steroids like cortisol. Arachidonic acid is generated. That's free arachidonic acid. And then it can react, usually after a CoA ester is formed, with cyclooxygenase, making the prostaglandins. That's the reaction. The COX reaction is inhibited by the NSAIDs, okay, like aspirin. Now, besides that cyclooxygenase, here we are in a little bit more beautiful detail for the LOX pathways. So here you go, arachidonic acid. You see it has 4 double bonds, 20 carbon fatty acid. This last double bond is 6 carbons away from the methyl terminus, so we'll call this an omega-6 fatty acid, which is what it is. What's going to happen if it reacts with either the 5-lipoxygenase, the 12-lipoxygenase, or the 15? It's going to depend on which carbon you're going to get that addition of oxygen. We're only interested right now in the 5-lipoxygenase, and that's where you're going to get this hydroperoxy residue now loaded up because that's where that carbon is at the 5 position. Okay? Now you're going to generate, at the first reaction of the 5-LOX pathway, 5-hydroperoxy icosahexatetraenoic acid. See, 4 double bonds. Now, what's going to happen is this is going to be removed, and you're going to get 5-hydroxyeicosatetraenoic acid. So this double bond is shifted. Okay. And you can see the other products depending on the other lipoxygenase pathways. Now, the 5-hydroperoxyeicosatetraenoic acid then, right, is you see how you get this epoxide formed. It's a spontaneous event carried on the LOX enzyme surface. This epoxide is very unstable, but this is LTA4. Believe it or not, LTA4 does have a residence time because there are proteins that bind to it, okay, and therefore maintain its structure. At any rate, if you add water to this system, okay, there's your conjugate triene, by the way. If you add water to this system, you're going to move around the double bonds a little bit, you see here, and you're going to leave a hydroxy group. And this is just showing you the actual reactive mechanism. Okay? That generates LTB4. There it is right there. So you don't no longer have really a conjugated triene system here because you've gone from all these bonds, you've changed the geometry of that bond there, and you've moved it one over. It's often what happens when you do either kind of oxidation or reduction, or simply the addition of water will do the same thing to these preformed conjugated double bond systems. So LTA4 again can also work with reduced glutathione, and there's your peptidoleukotriene that we talked about already, LTC4. So there's LTC4. Again, if you want to use these leukotrienes, peptidoleukotrienes, for example, these are the potent molecules in asthma and in shock reactions. Just removing components of that tripeptide, you make LTD4 and LTE4. See, there it is, simply with that sulfur bond there to the carbon atom. All right. Again, there's 5-lipoxygenase, there's 5-hydroperoxy. There it is, hydroperoxy. You get 5,6-epoxy-15-eicosatetraenoic acid. That generates then LXB4 or the LXA4. Okay. So these are known as the lipoxins. This is a different pathway, okay? And the lipoxins are yet another component of the repertoire of eicosanoids that are involved in pro-inflammation, depending on the cell type. So now we know where fatty acid comes from. We know what eicosanoid is. We know what LTB4 is. Let's now turn to the receptor, of course. The LTB4 receptor is called the BLT, okay? The BLT is a high affinity for its ligand initially. BLT1 promotes the activation and migration of phagocytic and immune cells, such as neutrophils, eosinophils, dendritic cells, like we talked about at the beginning, as well as macrophages, B cells, and T cells, so also lymphocytes, you see. BLT1 is indeed a G protein coupled receptor, so all the things we talked about with GPCR apply and affirm here, and activates at least two different G alpha family members, including the pertussis toxin sensitive GI0, shown here, right? So that is sensitive to pertussis toxin, and the PTX pertussis toxin insensitive GQ alpha proteins are shown here. Shown here is G alpha 1,6, right? Now, see, they do different things depending upon the moiety then of what kind of G proteins you have. There are different family members. So it gets always more complex when you start getting intracellular. LTB4 now could be in circulation or be secreted and be generating that increase in concentration that is, again, not a standard reciprocal of the distance, square of the distance away from the concentration, but it involves all those other parameters we talked about in terms of its potency. But LTB4 binds to its receptor, depending on if it's at the G alpha I, that's going to cause the inhibition of adenylate cyclase. You're going to tank cyclic AMP levels, so everything that's turned on by cyclic AMP is shut down. And if they're this particular cell type, you're going to start chemotaxing. And that chemotaxis of, let's say this is a neutrophil, is inhibited by the pertussis toxin. Okay. Now, this is the other pathway. Now notice that that's what the G alpha does, causes chemotaxis. The G beta gamma, that dimer stays together, will also induce chemotaxis via activating the phosphatidylmethylmalonyl 1,3 bis kinase, but it also activates that same moiety, the protein phospholipase C Phospholipase C is going to generate from PIP3 the inositol triphosphate, the sugar. The sugar is going to bind to its receptor, the IP3 receptor on the ER membrane, releasing intracellular calcium stores, and that's going to ultimately cause enzyme release. That's the degranulation of these particular innate cells. Okay, so it's going to these are called granulocytes because the granules are full of enzymes. Those enzymes are going to be secrete out after you generate this calcium increase intracellularly. Two things then: chemotaxis and degranulation. Okay, and calcium is necessary. You can get calcium directly via outside the cell, or you can get it from internal stores, and that's the way it normally works. Okay? And maybe even the BLT is signaling to open up that calcium channel. Probably is through sodium-potassium interactors. Okay. So the activation of those G-alpha proteins by BLT results in a phosphoinositol hydrolysis, intracellular calcium mobilization, and a decrease in cellular cyclic adenosine, cyclic AMP, followed by chemotaxis and degranulation, as I said. Like other GPCRs, BLT1 is phosphorylated in its carboxy terminus. That's what's found in the cytoplasm after it's activated. That means after ligand binding. So that's what we're showing you here. So you got a membrane phospholipid. Phospholipase A2 generates arachidonic acid. 5 LOX going through the pathway making LTA4, which can go through, depending on the cell type, the peptidoleukotrienes, or an R system here, because we're talking about the circulating leukocytes, LTB4. LTB4 now can be secreted and work back on epidermal cells, okay, where it can actually bind to the BLT2 receptor, carry out functions there, and look also that there is a COX product from the thromboxane synthase pathway, which will also bind to that receptor, so you'll get co-opting of that receptor. But here we're talking about BLT1, so you're going to be binding to the receptor on immune cells, dendritic cells, and things like neutrophils and macrophages, okay? So the production of the leukotrienes and the prostanoids and this 12-HAT, which is this unusual prostanoid, the arachidonic acid is cleaved. You make these different eicosanoids. LTB4 serves a high affinity ligand for BLT1 and induces chemotaxis and activation of various subsets of leukocytes. BLT2, shown here, originally identified low affinity receptor for our friend LTB4, the leukotriene, is actually a high affinity receptor for the 12-HHT, which is, of course, from the COX pathway. And what it does is enhance epithelial barrier function. And the enzymes you can see are named down here. This comes from seminars in immunology from last year. All right. From the same paper, now we're looking at the lung system. Okay, so remember I told you that these leukotrienes are the potent vasoconstrictors which are involved in acute asthmatic attack. Now, this is a different system than what our papers emphasize. We just want to show you the biological significance of leukotrienes. Okay. So, first of all, you get leukotrienes being generated like LTB4 from a mast cell, macrophage, neutrophil. You make LTB4. It binds to its receptor, right? So when it binds to its receptor, either here this could be a lung dendritic cell, or it can bind directly to this immune cell response here. So either way, you get dendritic cell binding. Let's look at this. You get the production of that chemokine, CCR7, that causes activation of the dendritic cell and migration to a regional lymph node. And look what it's doing. You've now mobilized the lung dendritic cell after binding to leukotriene B4. You've mobilized it, activated it, and sent it to the lymph node. Once it's at the lymph node, the major histocompatibility complex, or HLA complex on the dendritic cell, interacts with the T cell receptor, priming that T cell in coherence with an antigen, okay? And that's going to now take naive CD8 T cells and generate now this immune cell phenotype, which also has a BLT1 receptor, you see. And now we have a CD8, okay? And now that is a T effector cell. So this was just a T cell, a naive CD8 cell. Now we have a T effector cell. So we've activated it and differentiated what goes on inside that lymphocyte. That then migrates to the airways. This is all in the lung, remember, generating the really potent pro-inflammatory IL-13. And then you get airway inflammation and mucus production. This is the acute asthmatic attack. And this cycle just continues, you see. All right. So let's talk some specifics, some brass tacks about the paper that we're looking at. The experiments are designed to interrogate the amino acid sequence of that BLT receptor, responsible for both basal, which is constitutive, and ligand-induced, which means after the LTB4 binds, phosphorylation of the human BLT1. The methods include phosphotag, sodium dodecyl sulfate, PAGE electrophoresis, it's polyacrylamide gel electrophoresis, where there is a compound put in the gel that specifically retards any proteins that have phosphate, alkaline phosphatase, removable phosphate on a protein. And so that small molecule is actually melted into the polyacrylamide gel, and it will retard the protein's movement in that electrical field, in that SDS PAGE gel. And the reason that's important, you can separate the different phosphorylated forms of the receptors, because it retards the phosphorylation state of the protein. Of course, depending on how many phosphates there are, it's going to retard the mobility of that protein slightly differently, so you'll get a ladder effect, and you get production of a lot of phosphointermediates for that one BLT1 receptor. That's what it's all about here. So you have they also did site-directed BLT mutagenesis. What were they doing there? Well, what do you think? They are site-directing a modification of the amino acid residues at the carboxy terminal, or sometimes also in the loop structure of that protein, that BLT1, so that you get rid of the serine and threonine, which can be phosphorylated, and you just bulk them up into alanines. They can't be phosphorylated. Okay, so that's what was done. They did site-directed mutagenesis on that protein, and then they looked at phosphorylation patterns, both constitutive and hierarchical, subsequent to LTB4 binding. Okay? So they wanted to detect hierarchical patterns. They want to know what is the complete discrete nature of the kinase cascade on the BLT1 receptor, so that you get the refined chemotaxis and degranulation of these circulating leukocytes, and ultimately also the activation of the lymphocytes, particularly T cell lymphocyte, which is generated with CD8 positive T effector cell. All right. So the readout in this experiment of the mutagenesis experiments, right, altering those amino acid residues, included chemotaxis and also the release of one of the enzymes during degranulation. So the granules, when they're degranulated, release enzymes like beta-hexosaminidase, which of course is an enzyme that's going to start tearing down things that have hexosamide bonds. Hexosamide bonds are found in certain membranes of certain bacteria, okay, and also in some host cells. And anyways, that release is all going to be elicited by varying the concentration of LTB4. The results may suggest from this paper the imposed increasing LTB4 gradient as generated by the system we just talked about, phospholipase A2, LOX5, that particular gradient effect, enhances the BLT1 phosphorylation in a stepwise manner, and that all facilitates the precise migration. Because think about what's going on here. You've got these happy-go-lucky cells moving in the bloodstream, right? You've got dendritic cells, you've got neutrophils, you've got other kinds of monocytes, you've got monocyte macrophage lineages. All of a sudden, there's some damage to the system, and there's a signaling that's systemic, right? It's in the blood. How does that happen? Well, one of the ways is by generating this gradient of leukotriene B4. So what they want to know is, what is the precise migratory pattern now of these now activated, chocked full of enzymes to deal with the inflammatory response and to kill anything that's there, hence the pro-inflammatory cytokines and chemokines. And eventually T cells, T effector cells, and natural killer cells also play a role here, to kill or to destroy whatever the inflammatory response is and get things back to normal. All right. So anyways, these are all working at the local level, and that's what degranulation is. And that's a hexosaminidase release is actually the readout from Here's some data. Here's some really cool data. Remember, these are STS page gels. They're using these are immunoblots, and they use the immunoblots because they tagged with the hemagglutinin protein. That's the influenza hemagglutinin protein. This BLT1. They previously showed that when they tag it with that peptide fragment, so they can pull it down with an antibody to the HA tag, that it doesn't affect its biological activity. Particularly, you wouldn't want something that you tagged covalently with an HA tag to modify phosphorylation states, right? Because then all the data would just be what? It would be totally dependent on the HA tag. So they've already done those experiments to prove it's not. In fact, part of it's in the supplement of the paper. So they have a mock transformation, okay? So they have HeLa cells, they're working with cancer cells, HeLa cells, and they're expressing this protein they manufactured. So it's hemagglutinin tagged BLT1, okay? And you can see here that there's a mock transformation. Do you have any of this protein there when you pull down with HA? No. That's good because it shouldn't be there. Of course, when you do the transformation with this gene in the HeLa cells, yeah, you do get protein made. And look what happens. If you add LTB4, you get a different pattern, right, of phosphoproteins with and without the LTB4. Right? This is now just the immunoblot. And now here it is when you're in the phosphor gel, right? When you can really separate all those bands and getting separate. See how they're all clumped together here in just the immunoblot? Holy smokes, you add that you add it so that you retard the movement of the phosphorylated proteins, and now you get a nice laddering effect. And you can see here that without LTB4, right, you get a concentration of three main types, maybe, right? But when you add LTB4, you get a whole lot more of a variation of phosphorylation. That's all it's saying. All right. So that was all figure A. Figure B, the HeLa cells expressing that construct were stimulated with LTB4, and membrane infections were treated with CAF intestinal alkaline phosphatase before the STS page and immunoblotting with the tag. And what they're showing you here is when you add that alkaline phosphatase enzyme, you get rid of all those phosphoforms, you see. You generated them with LTB4, see, holy smokers. You now clip off that phosphate. It's called alkaline phosphatase because it works in the alkaline pH range. You get rid of all those intermediates, which tells you that that's indeed what's going on. Figure C is just the HeLa cells expressing the wild type or a form of the BLT lacking an extracellular and glycosylation site. And that's what that ON is, right? And here you're just looking at the different patterning that might occur, whether or not you have the wild type protein with or without LTB4, or you have the glycosylation site altered form, okay? We wanted to know whether or not that extracellular glycosylation site is going to affect the patterning, right, on this phosphogel here. And indeed, it does show that there's more of a concentrating effect. There are less, there might be the same number of bands, but it tends to be a dilution of some of those major bands when you're looking at the one that's lost its glycosylation site, but not very significantly, okay? And that's what D looks at. You're looking at HL60 cells, different cell line, and they're cultivated in the presence of all trans retinoic acid. It's been known that retinoic acid also is involved in stimulating LTB4 production. And in feed, you do see that all trans retinoic acid has a tremendous effect on that phosphoprotein ladder, right? So once you add the all trans retinoic acid as opposed to without it, right? And you're also adding LTB4, and here's the enzyme which is going to remove, right? That's the enzyme that's going to remove the glycosylation pattern. You get a significant shift. So what this all shows, this whole figure, is that LTB4, the icosanoate, the leukotriene, stimulates a set of membrane-associated phospho-BLTs. Remember, that's what these proteins are. They are the receptors that are glycosylation independent somewhat, but they are all trans-retinoic acid dependent. Okay, so there's more detail there than I'm going to tell you, but that's just giving you more of the fine grain of the co-stimulation, whether or not you need glycosylation patterns on these proteins. for them to be functional. Now, here's another really big slide. I love these papers from Science Signaling because they have so much information. So we're going to go through this quickly. This is just looking at the protein, kind of like a schematic. These are showing you all the serines, see the S, where they are, the positions of the cytoplasmic tail, that is the amino acid residue position, or the threonine residues. Here are all of the different changes they made. Serine to alanine, threonine to alanine. Here's another serine to alanine. Now, unfortunately, they labeled these. When I first looked at this, I said, why are they doing this? IL-1, IL-2. These are not cytokines, okay? They're just a label of the different mutants they made. So these CTs, for example, here's your threonine change. This one has a serine change, two serine changes, and one threonine, one serine, one threonine at those positions, and you can see those positions locked in and loaded over there. So what is all this, okay? You're looking at conserved serine and threonine residues in the cytoplasmic domain. That's what's down here. Now, this is a protein which has these 7 transmembrane spanning. This is typical GPCR protein. Okay, but all the activity we're looking at is down here in the cytoplasm. Okay, and notice that's probably why glycosylation didn't have an effect, but that's why retinoic acid might have, for reasons that have to do with whether or not glycosylation has a major effect in cytoplasmic components of proteins which are erstwhile membrane bound. That's really basically what that story is. All right, so just showing you the mutants they made. The B is looking at those mutants, okay, and expressing them in HeLa cells and the membrane fractions, and just showing you that they have different patterns in this phospho-retarding gel. These are whole cell lysates, okay. There's beta-actin showing you. This is just the control loading for the immunoblot. Okay. All right. Okay, C are HeLa cells expressing the indicated HA-tagged receptors, and whether or not they're treated, which is 100 nanomolar, or pre-treated with leukotrienes. You're seeing whether or not leukotrienes have a change in the phospho pattern, and some do and some don't, right? So here you've got the mock, there's nothing there. Here you've got this tagged one that we first looked at. You see the difference, whether or not you had LTB4, just like the past. Remember, these are immunoblots. Here's where you've changed all the residues. You have a tremendous effect. There's nothing when LTB4 is gone. When you add it, you get a tremendous increase in the ladder formation. And here are some of the other individual mutants, okay? All right. D is just showing you the indicated mutants and where they're constructed, where they are in the protein, okay? And that's what basically is just showing you a more fine grain of that figure. All right. E is looking at that mutant form of the receptor and all the indicated mutants expressed in the HeLa cells. Okay, again, showing you what they look like in terms of just the native state. They differ in mobility, okay? And the last of these STS page immunoblots is the membrane fraction, okay? That's where the protein is, right? Again, from HeLa cells expressing the indicated forms of the BLT treated with 10 or 100 nanomolar. They're showing you whether or not there's a concentration effect of the LTB4. Now, what all this data says, just jump to the quick, is it's a substitution of all 5 residues of BLT1 ON, that's the delta B, that's all 5 residues, that's what that is, led to the complete disappearance of shifted bands in the absence of LTB4. Okay, see, complete absence. You see that? There's no more shifting. You got rid of all of them. That's what that meant. So the BLT delta B phos mutant means everything's gone, completely devoid of the basal phosphorylation, will display an LTB4 stimulated band shift. That's in 2F. So even though you got that, you do get a band shift when you add LTB, because those aren't the basal phosphorylation sites. Those are somewhere else in the protein, the hierarchical patterning subsequent to the ligand binding, you see? Demonstrating the basal phosphorylation wasn't required for the LTB4 induced phosphorylation, separating basal phosphorylation to LTB4 ligand inducing phosphorylation. The human BLT1 has 5 basal phosphorylation sites. There they are. There are 1, 2 serines, 3 serines, and 2 threonines. And there's the position on the polypeptide backbone. And there are 2 LTB4 induced phosphorylation sites. That's threonine 308 and serine 310. and that's all in the C-terminal region of the polypeptide. Right? Phosphorylation of these residues may occur in many different combinations. That's basically what the data is showing. Now, there's an enhancement of the BLT1 phosphorylation by ligand in a dose-dependent manner. So as you add more of the ligand, more of the activating molecule, right? You're titrating now the phosphorylation. Remember now, this is the receptor that doesn't have any of those cohering basal phosphorylation sites. So all of this is just totally dependent on LTB4, and you start increasing those LTB4 dependent phosphorylations. There's serine 310, threonine 308, right? And then there is your stable serine 310, that just stays the same, right? That's what's showing you here. All right. Now, again, patterning. So these data mean that phosphorylation of each of the LTB4 induced sites could trigger additional phosphorylations at the sites initially identified as basal. Okay, so you're getting a retailoring even at the basal. So that means that we thought the basal was stationary, flat affect, but as it turns out, once you start triggering the LTB4 dependent phosphorylation sites, the receptor goes back and alters the phosphorylation pattern of those original basal sites, right? Now that tells you that you have a basal activity, then you have an induced, then you have an altered basal, then you have an altered basal and induced. It's going to be 4 different structures of these receptors. And that's going to precisely give you no chemotaxis, no degranulation, granulation off. See, that's how it functions. Going back to number 1, right? 4 different conformers of the BLT1 depending on phosphorylation state. It's beautiful. So here's a protein. Here's your basal phosphorylation. Here you're getting more phosphorylation of the sites, you see here. As you increase the gradient of LTB4, you get more and more. So there is an abrupt feedback control, as I mentioned at the very beginning of my talk, over the LTB4 mediated activation of its receptor. This is associated with increases in intracellular calcium, which we saw before when the bradykinin bind or the TNF alpha bound, and they're immediately shut down. Okay, all of this alteration in phosphorylation after the initial stimulation when you pre-stimulate with a very low amount of LTB4, 10 nanomolar, very low. By the way, these icosanoids work at absurdly, insanely low concentrations. That's why they're so potent. Exposing cells to 100 nanomolar, which is still really small compared to a lot of metabolites in the cell, of LTB4 after stimulation with 10. So you incubate with 10, then you pre-stimulate with 10, then you give it 100. Increases the extent to which the BLT1 was phosphorylated compared to the stimulation with the 10 alone. It makes sense. So from a basal state to an induced state. BLT1 remains on the cell surface all the time, okay? Thus can be modified by additional phosphorylation sites upon activation. So the absence of LTB4, the basal sites are phosphorylated. Low concentrations of LTB4, see, you start to up that gradient. You get phosphorylation of serine 310, which stimulates additional phosphorylation of the basal phosphorylation sites. Boom, 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 boom. Higher concentrations stimulates the phosphorylation of the threonine 308, which in turn promotes further phosphorylation events at the C-terminal region. You start altering now all these phosphorylation sites. This gives you a lot more of the fine grain. Okay. In the absence of LTB4, see, just your happy G protein coupled receptor BLT1, bound to GDP, so it's in its off state. You've got this 2 phosphates on that cytoplasmic tail. The basal phosphorylation sites are variably phosphorylated, yielding a mixture of phosphorylated species. Okay, that's what's going to happen. The receptor still isn't active, and now why do you have a mixture? It's because that's what's left over after this entire stimulation cascade, right? You're not going to remove them all, so you're going to have a mixture of them. That gives you further fine control over switching, moving chemotaxis and degranulation. I think it's absolutely amazingly awesome that you have all these different phosphorylation sites. You're controlling them at the basal level, but even the basal level has some previously altered basal level phosphorylation sites, which gives you then the nuanced structures within the given neutrophils and lymphocytes so that they can now change and move and drift. They can interact, for example, to the naive T cell, triggering it up, turning on a CD8 positive TF cell, right? At different levels of concentration and at different qualitative changes in chemotaxis and in degranulation. Just absolutely amazingly beautiful and stunning what the kinase cascade can do here. All right, so downstream GI protein is bound to guanosine diphosphate, right? Yeah. Low concentrations of LTB4. You got a high affinity receptor, very common when you have low levels of low concentrations of ligand. You have a high affinity receptor, okay? Stimulates it to adopt that conformation. So you're changing the conformation. That's what these phosphates are doing, by the way. They're just altering the three-dimensional molten structure of the receptor in the membrane. Okay. All right. And now you exchange up to GTP. Now you're loading it. You're triggering it, right? You're loading the receptor so that now you can do your job. You can move. See, you can start chemotaxing. You can start doing a cell response. So once you load GDP out and take, that's the exchange factor that takes GDP out, swaps it for GTP. Then you're going to get a hydrolysis of the bound GDP to go back to GDP, which is different conformer state because now you've got a low affinity. So let's go on here, okay? So after primary signaling, the GI exchanges GDP for GDP, or you hydrolyze it, okay? And serine 310 is phosphorylated, so it's left over here. Right? Additional phosphorylation of the basal sites then occurs, leading to ultimately a return to the low affinity type. Right? As neutrophils migrate up that gradient, you're getting more and more LTB4. Remember, as you get closer and closer to where the source was, where it was synthesized and secreted by yet other circulating leukocytes, right? As they migrate to that region where presumably where the lesion happened, where the damage occurred, higher LTB4 concentrations stimulate the phosphorylation of the threonine 308. Okay, here comes all the complex detail we got here, which in turn then promotes an additional phosphorylation events shown here. Okay, and that ultimately leads to secondary responses such as degranulation. So chemotaxis, stopping, concentration of LTB4 is higher, degranulate. Okay. So this is a beautiful system, like I said. Ultimately then, you got the neutrophil, a chemotaxis because of its phosphorylation pattern. You go from a low concentration to a high concentration of LTB4. Carrying out the normal chemotaxis, you then degranulate at the site where the highest level of LTB4 is, presumably where the lesion is, where the damage is, where the inflammation is. So highly phosphorylated BLT1 is important for the precise directionality of chemotaxis and the degranulation at the higher LTB4 concentrations. The absence of those phosphorylations results in an aberrant migration pattern and basically the dead end. That was the delta phos, where you got rid of all of the constitutive kinase phosphorylation sites, Okay. B then shows you not only is that happening, how does the actual chemotaxis happen? Oh my God, this is so cool. This is the schematic representation again. Now, these are different proteins that are going to be involved in tailoring the phospholipids that are actually within the leukocyte. Okay? So now you have the BLT wild type versus this one that's been altered, right? Yeah. Either way, a gradient of LTB4 is going to cause a movement of phosphatidylinositol membrane lipid. You're going to start polarizing this cell with membrane lipid, generating a leading edge by putting out more PIP3, PIP345 trisphosphorylated inositol phosphate. that's going to cause the mobility to go in that particular direction. And so this is the P13 kinase dependent phase because you've added phosphate to those lipids, right? So remember way back when we talked about how the phosphoinositol kinase cascade is also involved? It's involved in that calcium release and the chemotaxis and degranulation. It's also involved in bulking up, building up a higher concentration of the phosphoinositide lipids at that particular leading edge, causing migration to move that way and no other way. Outrageously cool. Okay. So that's what this says here. During initiation of chemotaxis, neutrophils become partially polarized without this lipid, the phosphoinositol 3,4,5 trisphosphate, and they're able to migrate with correct directionality. Thereafter, this P13 kinase gamma form and the P10 and SHIP1 cooperatively localize those lipids, okay, because these are enzymes that work on the phospholipid, to the prospective leading edge, like I just told you, and that process then leads to the assembly of the leading edge and full polarization of the leukocyte, both of which are critical for proper directionality and therefore chemotaxis. The impaired directionality, which is down here when you got rid of all of those basal phosphorylation starts, you don't respond to the gradient of LTB4, you get a dead end. Things don't work. You don't get control over the inflammation. And that's probably due to the reduced production of the phosphatidylenoacetol 3,4,5 trisphosphate. Results in the loss of that accumulation at the leading edge, where the LTB4 concentrations are at the greatest, and that's where all the business end is going to be functioning anyway. Okay? Absolutely excellent paper. So let's discuss it finally. As neutrophils migrate toward the source of wounding or stress, the lipoxygenase 5 mediated increase in the concentration of our friend, the leukotriene B4, to which the BLT1 is exposed, gradually is going to increase. This induces the mechanism of enhanced and hierarchical receptor BLT1 phosphorylation at those serine and threonine levels, transforming the receptor now from a receptor to an LTB4 sensor. So the receptor molecule becomes a sensor molecule, sensing the relative gradient concentration of the highly potent hormone LTB4, attracting, causing chemotaxis of those cells, ultimately degranulation, dependent upon the concentration of LTB4 generated from the previous axis of site of injury, where there are already neutrophils hanging around and probably T cells starting to be activated. So there's all kinds of other cytokines being functioning, and the whole TNF alpha system is going on there at that site. So these neutrophils migrate toward the source. BLT1 becomes progressively more phosphorylated. We just saw that, and switches from the high affinity for the ligand to the low. Okay, and that causes then degranulation. Once exposed to a high concentration of LTB4, high enough, okay, it finally activates the low affinity form because it's low affinity, right? You got to have a whole higher amount. This is the ultimate signal. Like, is it okay to release these enzymes, to degranulate? Is it okay? Is it okay? By God, it is okay because the concentration is so high because we're at the source of where it's being synthesized via the PLA2 LOX5 pathway. Triggers other cell responses, and that's degranulation. So the enzyme is released. So polarization of the neutrophil, which we saw through that phosphatidylenoacetol tris kinase pathway, instantiates the chemotaxis, requires sufficient trisphosphorylated, as I said, inositol phosphate in the leading edge of the cellular membrane. And that basically demonstrates this whole thing, how lipids mediate the immune response without nascent gene expression. You don't need any other gene expression here. Once you've got the LTB4, the BLT, all subsequent effect happens once you get there. And that means the release of pro-inflammatory cytokines, growth factors, stimulation of the T cells, activation of the MHC complex or the HLA complex, etc., etc. Lipids are doing all this work, and all you're doing is moving them around and hydrolyzing them, or in this case, oxygenating them to the LTB4. Absolutely stunningly cool way of controlling at the exact position of where there's the site of inflammation, like say a wound, where you need to have the highest concentration of LTB4 to get those neutrophils there, chemotaxs there, release their enzymes, degranulate, and then start functioning with dendritic cells, interacting with them, and they're also chemotaxing there, ultimately working with the T cells to generate effector cells, which go on then to bring on the acquired immune response and totally take care of, clean up that inflammatory response. All the time keeping a feedback control mechanism by that 4-stage change in the overall phosphorylation state of the BLT1, going from no affinity to low affinity to high affinity, and then ultimately LTB4 gradient regulating whether or not you degranulate. Stunningly cool paper, and hopefully you get a real glimpse of what's going on at the submolecular level, subcellular level, when you talk about a simple thing like an injury, like a wound, some peripheral of the body. Same time this is happening, you're also getting pain mediated responses, right? And we told you that has to do with the cyclooxygenase pathway, right? Producing compounds, cyclooxygenase products, which are transducing through that afferent pathway, ultimately to the brain to cause the pain response, nociception. Just absolutely stunning the way all these things fit together. Anyways, thank you for your attention. That's me, Dr. Dan Guerra. There's the email address of our company. Contact us directly through there and become our client. We're glad to hear from you. Here's our website. Go there and you can read a whole lot more about us. Remember what we are in VerEvMed. We're a company where scientists verify published evidence in the medical biosciences. Verification of evidence in biomedical research literature. That's what the VerEvMed stands for. This is the Continental Divide in Colorado. Okay. Oh, and by the way, this first picture, this was, remember what kind of tree this is? I've talked about it many times before. I wonder what kind of wood that is. Yeah, that's a white fir. That's a dead white fir tree. And this is where I dropped it and where I was starting to cut rounds with my 044 steel chainsaw, which I bought the year my older daughter was born, 1988. This chainsaw is going to be 30 years old and it's still working fine. A tremendous piece of equipment. And that's a 28-inch bar, so I can cut these big trees down. All right, enough of my chainsaw. It is really a cool picture, though. All right, so the only thing left to do is to give you my sign off, and everybody knows what that is.